Hello friends, in the series of cell division video we had already talked about the mitosis, the one phase of karyokinesis, which has got four phases, of which we had discussed about two first phases, okay, the preliminary phases, that is the prophase and metaphase, right? And this video we will begin our discussion with anaphase. So what happens in anaphase? In metaphase, what happened? From both the centriole through the spindle fiber, the chromosomes are attached at the point of kinetochore. Now what happens is the spindle fiber will retract. That is what is going to happen in anaphase, and you could see, right, this, okay, you can consider this blue chromosome here, which is at the end, is getting split into two. That is, the centromere itself will divide, right? Centromere itself will divide. See, let me describe here. Now, if this is the case, right, and you have a centromere here, what happens? There is going to be a split in the centromere, thereby the one chromatid is going to become two chromatid, right? Let me call this as A, let me call this as B. Now this is A, this is B, because due to the pull from the spindle fiber, right? Now friends, if you look at my discussion, how do you count the number of chromosome, you would know that you do it actually by counting the number of centromere, right? Now this was actually two chromatid, one chromosome, okay? Why one chromosome? Because there was one centromere. Now due to the split, the chromosome number itself is increased. That is, here it is going to be two chromosome. Hope you get my point. This is not just two chromatid, because you could see there is one centromere here, there is one centromere here. So it is going to be two number of chromosome itself, right? It's going to be two number of chromosome. So this event, important event, that is, my point is the dyad chromosome, okay, when the spindle fiber pulls the centromere, the dyad chromosome, it's split into two individual monad chromosome, right, by a division happening at the mid of the centromere, right? Because this is very important, because when you solve problems in cell division, either for your competitive exam or for whatever purpose, this understanding this concept is very much required for you to have a clarity and easy approach in solving those problems, right? So this is the case here. Likewise, you could see that the chromosome itself is split into two, right? And the other thing happens is, once the chromosome is split, it moved towards the opposite pole. You could see that, okay, the chromosome moved towards the opposite pole, okay, because the spindle fiber starts pulling the chromosome, okay, towards its respective pole. And in that movement, you could see that the centromere is towards the pole and the chromatid is towards the equator. This is going to be the equator. Equator is an imaginary axis, not any physical structure, right? So this is the equator. The same cell is what is depicted here. This is the equator. So the chromatid is towards the equator and the centromere is towards the pole, right? You call this as a leading axis, this as a trailing axis, this as a trailing axis, right? And this as a leading axis, right? That is, the chromatid is going to be the trailing because it moves last towards the pole, and this is going to be the leading axis, right? So this is what happens in anaphase. So here there is going to be a split of chromosome, okay? So if there is going to be 23 chromosomes, it should be 46, right? Or if it is going to be 10 chromosomes, it should be 20, because there is going to be a split in the centromere, right? And the last phase is going to be telophase. Always telo means last. What happens in telophase is going to be exactly the reverse of prophase, right? In prophase, what happened? The chromosome condensation. So it's going to be decondensation. And here the chromosome is found as a mass, entangled mass, rather than a distinct compact structure, as you see in metaphase, late prophase or metaphase, okay, the reverse. And then you could see that the nuclear membrane begins to reappear again, where you could see the nuclear membrane disintegration in, okay, at the beginning of mitosis. Here the nuclear membrane reappears, right? And the chromosomes, two set of chromosomes are found as a mass towards the poles, right? Okay. and all the organelle which had disappeared like endoplasmic reticulum Golgi complex starts reappearing, right? So this is going to be the mark the telophase of the cell division, right? So now what actually you see is within a cell there are two nucleus, right? You can even call this as a dikaryotic condition, two carry-on nucleus, dikaryotic condition. Now we expect a split at the center, which is what is called as a cytokinesis, because this is the cytoplasmic part. There is going to be a split at the center, which is called as cytokinesis. Now this is going to make this part into this cell and this part into this cell. So that is what is we are going to discuss in cytokinesis. Actually, cytokinesis we will make it as a separate discussion, because it has got some informations to be discussed. I am not going to include here, but cytokinesis is generally varies from plant cells and animal cells, right? It's not going to be the same, because the basic outlay of the plant and animal cells are going to be different, because the plant cell has got an extra membrane which is called a cell wall, and that is going to make the cytokinesis bit complicated, whereas animal cells don't have a rigid cell wall, whereas the cytokinesis is going to be somewhat easier. So let's discuss that in cytokinesis, which we'll do it as a separate discussion. Friends, I hope you get an overview of what happens in mitosis, right? So you could see that when you compare the parent cell, whatever the chromosomal composition you see here, the same thing you should be able to see in both of the cells. You see there are two important outputs that you are supposed to note with respect to mitosis. That is, there are two daughter cells which are formed, whereas in meiosis should be 4, here 2, and this two daughter cells are going to be exactly same among themselves, and also when you compare those with the parent cell. Hence mitosis is what is called as a equational division, right? So this is what regarding mitosis. So this understanding is going to help you in solving problems, and also understanding the basics of mitosis also helps you in having a clear-cut clarity understanding in meiosis, right? As I said, meiosis is going to be as same as mitosis. Here and there is going to be some differences. You see, you're going to see only one major difference in prophase 1 of meiosis. Rest of all is going to be as same concept that is followed in mitosis. So kindly go through this video. In case of any doubt, please do not hesitate to get it clarified. So let's meet in another video with meiosis. What is meiosis? How exactly meiosis is going to differ from the mitosis? And then we conclude the series of video with cytokinesis and other trivial factors there in the cell division, right? Thanks for watching.